Good morning. I'd like to thank the organizers today for giving us this opportunity to present this talk on our thermostabilized ricin toxin vaccine, which we are developing under the animal rule. As a public company, we have to make certain precautionary announcements about any forward-looking statements we make. But actually focusing now on the subject of the talk, what is ricin exactly? The ricin toxin is actually a very potent toxin with extreme toxicity, especially when given by injection or inhalation. The ricin toxin itself is actually made up of two parts, what's known as the A chain and the B chain. The A chain is actually a ribosome inactivating protein, and actually as little as one molecule of RTA in the cell can completely shut down cell protein synthesis, which is clearly fatal to the cell. The B chain then is primarily important in delivering the A chain into the intracellular part of the cell. The symptoms of ricin intoxication are generally driven by where you are first exposed to the ricin. So, of course, breathing in the ricin results in significant respiratory distress immediately, whereas ingestion might result in gastroenteritis and inflammation, and injection may lead directly to multiple organ failure. Regardless of the route of administration, lethal effects are usually observable within 48 to 72 hours, and more importantly, irreversible effects become apparent within about 4 to 12 hours. Ricin is considered a significant biothreat, and the reason for this is just primarily related to its ease of production. Ricin is produced as a byproduct of castor oil production, and there's 1 million metric tons of castor oil made in the world every year. There's been a history of suspected stockpiles and use of ricin intoxication, not only in war scenarios, but actually more recently in the news where people attempted to mail ricin products to the Pentagon and the White House. As I mentioned earlier, some of the most disturbing aspects of ricin is that the onset of toxicity is very rapid, and it's also rapidly irreversible. Moreover, even if ricin doesn't cause mortality, it can also cause significant long-term damage and morbidity. Because the treatment window for reversing ricin intoxication is so very short, a preventive vaccine strategy is clearly superior to most post-exposure approaches. However, at this point, there's actually no approved prophylactic or post-exposure therapy for ricin intoxication. Sologenics then is developing a ricin toxin vaccine, which we call Rivax. And actually, it's the antigen to Rivax is very simple. It's actually just the A chain mutated in two specific spots, which obviates all known toxicities. We've actually demonstrated the safety of this antigen in two different phase 1 studies, one with the antigen alone and one with the Alum Adjuvanted antigen, and we'll discuss that in a little bit more detail. Subsequent to these phase 1 studies, we've actually worked a great deal with this subunit vaccine to enhance its stability. We've used a proprietary thermostabilizing technology that allows us to lyophilize this subunit protein despite the fact that it's adjuvanted with alum. As many of you will know, alum adjuvanting of subunits has traditionally been a problem precisely because it causes a stability issue which cannot be resolved by lyophilization, and we've overcome this problem by incorporating specific generally regarded as safe or GRAS excipients. In fact, we'll see the data today that our final lyophilized formulation is stable for greater than 1 year, even when it's stored at 40 degrees Celsius. Another nice aspect of this particular formulation is that it's actually reconstituted immediately prior to use by simply using sterile water for injection, a very easily available commodity. Finally, we've demonstrated the efficacy of our Rivax vaccine in animals, both in rodents, where we've shown that it works irrespective of how the intoxication occurs, so by inhalation, ingestion, or injection. And we've further shown up to 100% protection of primates when they're exposed to the most lethal route of exposure, which is inhalation. So just to revisit the clinical safety data we have for Rivax, we've conducted, as I said, 2 phase 1 studies. The first of these was just the Rivax antigen by itself, with doses given on a monthly schedule for up to 3 monthly injections. The injections were very well tolerated. We did get immunogenicity. Both anti-ricin and neutralizing antibodies were detected. But as this was an unadjuvanted formulation, not surprisingly, the antibodies were relatively low and short-lived. The next step then was to proceed with an alum adjuvanted vaccine formulation. This formulation was given again 3 times on a slightly different schedule at 3 different dose levels, and the injections were very well tolerated. As expected in the presence of the alum adjuvant, the antibody levels were higher and longer lived than with the liquid formulation. However, one nuance of this particular study is that the alum adjuvanted formulation was manufactured immediately before injection to the healthy volunteers because of the known stability issues with the particular formulation. Clearly, while this was very feasible in a phase 1 study, it certainly isn't a feasible approach from the perspective of stockpiling and long-term use. For this reason, then, we had advanced a thermostabilized formulation, which we've spoken about earlier. And really the key to this thermostabilized formulation is the mixture of grass excipients with the alum adjuvanted protein, which enables it to be lyophilized without causing significant particle aggregation. As I mentioned earlier, this material can then be stored for extended periods of time before being reconstituted with water immediately prior to use. And here on slide 8, we have a presentation of exactly this data. So here we compared 2 things. We compared our lyophilized thermostabilized formulation, which we stored at 40 degrees Celsius for more than 12 months, and then reconstituted immediately prior to use. And we compared that to the liquid vaccine, which was made at one time and then stored in its liquid form at 40 degrees Celsius. And this liquid formulation is shown in red on this graph. Now we evaluate the stability of this vaccine by simply asking how well it protects mice who are vaccinated on days 0 and 21 and then challenged on day 35 with ricin injected into the intraperitoneal cavity. And what we can see is that at baseline, both vaccines were effective, the thermostabilized vaccine showing, as usual, 100% protection. However, as time passes with the vaccine being stored at 40 degrees Celsius, the liquid vaccine clearly degrades, ultimately losing all ability to protect animals by 6 months, whereas the thermostabilized vaccine maintains its potency. Now, as you can imagine, this was a fairly extensive data set, and we'll be returning to the immunogenicity measures that were also taken during this study in future parts of the talk. More importantly, perhaps, than showing protection with these vaccines in mice is showing that non-human primates, which have a similar, or a more similar at least, lifestyle to humans, as well as a more similar anatomy, are also well protected in this case. Here we see a situation where we have non-human primates that were vaccinated on a monthly regimen 3 times, so on days 0, 30, and 60, and then challenged with between 3 and 5 times the LD50 dose of aerosolized ricin on day 110. Note that this is aerosolized ricin, which is the most challenging route of administration to protect against, as well as the most toxic route of administration. Here we see that the unvaccinated control animals rapidly succumbed to intoxication within about 50 hours of breathing in the ricin. In contrast, the vaccinated animals clearly had no acute mortality whatsoever, and only 1 late death that was not directly attributed to the ricin intoxication. Again, this vaccine was very protective in this setting, further supporting the use of the Rivax vaccine. This was done with our most recent thermostabilized formulation, and the results have been subsequently published. One of the most interesting things about the program that we're working on is our ability to leverage some previous work which had been done by Dr. Nick Mantis and his colleagues at the New York State Department of Health's Wadsworth Center. Specifically, they'd done extensive mapping of B cell antibodies and where they bound to the ricin toxin itself, both when they're protecting and when they're not protecting. And as a result of this, there is a series of antibodies, monoclonal antibodies, which have very well-known epitopes and binding spots on the ricin toxin and are known to be neutralizing. From their work, they've identified 4 major areas or clusters where these antibodies bind to the ricin toxin A chain. And these are shown in this diagram. What's particularly relevant is 2 specific antibodies I'll be talking about in great detail in this talk. The first of these is known as PB10, and it binds in cluster 1, which is shown in the blue and teal color. The interesting thing about this antibody is that it recognizes a linear domain of the protein, which means it's a very robust recognition that works over a broad range of degradation states of the protein. In contrast, there's another antibody known as SYH7, which binds to cluster 2, which we see colored in orange. This particular antibody actually recognizes a 3D conformational state of the protein as opposed to a linear sequence, and is therefore more sensitive to conformational degradation of the protein. So I mentioned in the title of the talk that we're dealing with the animal rule as we attempt to develop this ricin toxin vaccine. Clearly, the animal rule becomes relevant when efficacy testing in humans is unethical or infeasible. In this case, clearly, we would not be challenging humans with ricin toxin. Under the rules of the animal rule, what we have to do is efficacy studies in animals to demonstrate that the material is protective, but safety studies are conducted in humans. Therefore, the animal rule really depends on well-characterized animal models, much more so than any other route of clinical development. There are 3 basic constraints that these animal models have to satisfy. Firstly, the disease pathogenesis in the animals must mimic that in humans. This is very straightforward in the ricin toxin case because ricin simply kills the first cells it comes into contact with. Secondly, the response to your drug or vaccine in animals must mimic that in humans. And thirdly, the response to the drug in animals should correlate with survival in animals so that you can predict survival in humans. It's worth mentioning at this point that these biodefense programs often qualify for orphan and fast-track designation. Rivax, for instance, has orphan designation from both the US FDA and the EMA. Under the 21st Century Health Cures Act, biodefense agents that are approved may also qualify for a priority review voucher from the US FDA. So to come back to those 3 main constraints I outlined for animal rule approval, the first is that the disease pathogenesis is consistent. Now, as I said, this is very straightforward in the case of ricin toxin, and unlike most other biodefense initiatives, because ricin simply causes cell death in whichever cells it sees first. This means that really the disease progression has more to do with the route of administration of the ricin than it does the particular animal species that have been exposed to the ricin. And while we certainly don't have very much information on human aerosol exposure, we have a fair bit of information about data between rodents and non-human primates, and there is significant similarity between these 2 species, supporting this argument about the consistency of the disease pathogenesis. Secondly, we have to demonstrate, of course, that the response to our vaccine is consistent across species. And this is where the work with the Wadsworth Center was really very valuable, because we could ask a very basic question. We could ask the question, how much do the antibodies in the sera of the vaccinated individuals, be they non-human primates or humans, compete with these different monoclonal antibodies for their ability to bind to ricin? If the sera competes with the monoclonal antibody, then there are at least some antibodies in the sera which bind to the same epitope. And this allows us, using multiple monoclonal antibodies, to generate an epitope profile of the sera. Here we can see that this work that was done both with the non-human primate study I showed you earlier, as well as the samples that had been previously collected in our phase 1b clinical trial, indicates that the two species have a very similar epitope profile in response to vaccination. Finally, we have to be able to draw some predictions about if an animal is protected at level X immunogenicity, so shall a human be. And this correlation is, again, an added level of rigor which is not normally seen in programs that are not required to proceed via the animal rule. There's a number of potential options for immune correlates of protection with a vaccine. These are fairly obvious. The first of them obviously is total anti-Ricin IgG levels. The interesting thing here with the work we've done is that clearly the IgG levels can be dose responsive as we increase the doses of vaccine, but there's limited sensitivity for protection to subsequent challenge, and I'll show you some of this data. We can also measure neutralizing antibody levels. These, of course, are cell-based assays, which inherently means they have limited sensitivity, and that's particularly problematic when you're trying to correlate with rodent data because of your limited ability to draw blood from rodents. And finally, we can rely once again on these very well characterized monoclonal antibodies, and in fact we can think of using them in two different ways. In the first way we think of using them, we can use the antibodies to bind directly to our antigen, which is just the mutated ricin toxin A chain, and ask if the protein conformation of our known epitopes are stable through time, and if their stability or lack thereof correlates with ultimate survival in a vaccine challenge model in rodents, for example. And we'll show you some of this data. And the other is to recapitulate that study I showed you earlier, where we asked if the antibody, monoclonal antibody, and the sera of a vaccinated individual compete with each other, and if the degree of competition can be correlated with survival in animals. In fact, both of these potential approaches work, as we'll show you in the upcoming slide. So starting then with the simpler approach, which is, can we interrogate the conformational stability of the protein itself and ask if any change in that protein stability then correlates with a change in survival. The real attraction of this particular approach is that it's something that can be done even though the protein is bound to the alum. And as many of us know who work with alum adjuvanted vaccines, it can often be very difficult to measure much things about the protein once it's bound to alum. So this is a very unique assay from that perspective. Here we're using that same study we've done with the mice that I showed you earlier, where we incubated the vaccines, either just the liquid formulation, which remained liquid, or the lyophilized formulation at 40 degrees Celsius. And what we see on the left-hand side is the liquid absorbed vaccine. Here we see that clearly protection is falling, so it goes from 75% down to 33% and then down to 0 by 6 months. And at the same time that our protection is falling, our ability of these antibodies to recognize those conformational domains is also degrading. So this is actually measured as an inhibition. And we can see that we're losing our ability to bind to the ricin toxin A chain as the protein degrades. In contrast with the lyophilized vaccine, we maintain survival throughout the 12-month period, and we also maintain our relative binding inhibition in competition with ricin and our ricin toxin A chain, indicating that the protein is remaining conformationally intact. The other way I mentioned interrogating these potential immune correlates of protection is looking at this epitope profile. So just to go over this assay again, what we're really doing is taking a designated monoclonal antibody, and we'll be talking in this case about both PB10 and SYH7, and we're putting it down on the plate. And then we're taking biotinylated ricin and incubating it with the sera from the vaccinated individuals. Then we ask whether our monoclonal antibody can still fish the ricin out of solution. If it can still fish the ricin out of solution, clearly our monoclonal antibody and the antibodies in the sera bind at different places on the ricin. However, if it can't fish the ricin out of solution, then clearly the two of them bind at in the same general vicinity. So this means that you can measure a percent inhibition of binding, which relates to the sort of prevalence of binding in the sera to that known epitope. Again, coming back to that same study where we looked at our vaccines that were stored at 40 degrees Celsius, and we also looked at them stored at 2 to 8 degrees for obvious reasons, and we asked what the comparison, what the immunogenicity readouts would be if we drew blood 5 days before we actually challenged the individuals with ricin via the intraperitoneal route. Now, as I mentioned, we do get increasing IgG, anti-ricin IgG titers as we increase the dose of the vaccine. And as you see in the top right-hand side of this slide, we also see significant IgG titers in survivors. However, the interesting thing is that there are some survivors who test with no detectable anti-ricin IgG level, yet still survive subsequent challenge with ricin. And similarly, there are animals that have detectable anti-ricin titers, but actually succumb to subsequent ricin intoxication. And the overlap here is a bit stronger than we would ideally like. However, when we look at these epitope profiles with the PB10 and the SYH7, as I mentioned earlier, we can see that there's a better separation between our decedents and our survivors. In fact, we can push on this a little bit harder, and specifically with respect to the SYH7 antibody competition profile, where we had seen the best separation between the decedents and survivors, we took just the mice that had been exposed at the time zero evaluation of the vaccine, and we developed a model, a binary classification model, that said that if the ability of the sera to compete with SYH7 came above a certain threshold cutoff, then those animals were most likely to be protected to subsequent challenge. And in fact, the overall model accuracy was very high when we developed that model. But of course, the true test of a model is what happens when you use it in a more predictive framework. And so we actually applied this same model on a forward basis when we did our 3, 6, and 12-month stability time point stations. Here we see the overall performance of the model in these remaining 546 mice, where the overall model accuracy was 74%. Moreover, we had very high accuracy predicting animals that would succumb to subsequent ricin intoxication. And from a clinical perspective, this is exactly the type of model we would want to have. Clearly, if we have an individual who is not yet adequately protected, we can administer a booster shot of the vaccine to improve their antibody response. And it's very important that we can identify those individuals. If we accidentally identify someone who would have otherwise survived anyway, and the consequence is they get an extra vaccine dose of what is a very safe vaccine, that's not really a very large concern. So we find this model compelling not only in its overall accuracy, but also in its basic characteristics. In fact, we've looked at the same epitope profile, not just in mice, but also in non-human primates. Now, clearly in the non-human primate framework, we have nowhere near the number of animals we do in our mouse studies. But this is the second non-human primate study we've done where, again, we did looked at exposure to inhalation around 3 to 5 times the LD50 for the non-human primate, with again the vaccination scheduled on days 0, 30, and 60. And again, that 100 microgram dose of the vaccine was 100% protective, so this was very consistent with our previously published work. We also found that if we significantly reduced the vaccine dose, then of course we reduced protection. And perhaps most interestingly, we found that if we only gave 2 vaccinations on day 0 and 30, we were still able to protect a significant proportion of the animals. Again, when we looked at these different immunogenicity readouts prior to challenging the animals with aerosolized ricin, we found again that the SYH7 really gave us the best correlative readout of survival above a certain threshold level. So in conclusion, I think I've shown you that our ricin toxin vaccine, or RIVAX, has demonstrated consistent efficacy in animal models and has demonstrated efficacy both when the ricin is introduced systemically as well as by aerosol or inhalation. As we've discussed, the ricin toxin vaccine will need to be approved via the animal rule, and we've shown that our disease pathogenesis is similar, that our vaccine response is similar, and moreover, we've identified correlates of immune protection that we think will be significantly helpful in the clinical setting. RIVAX itself has already been tested for safety in humans, and the preliminary formulations demonstrated very good safety and immunogenicity. We do have an upcoming phase 2 study planned where we are planning to look at new immunogenicity markers with our new thermostabilized formulation. One of the interesting aspects of this program has been how we can interrogate the quality of the immunogenic response with monoclonal neutralizing antibodies, and it is worthwhile to think about whether this approach could have utility for other vaccination programs. I'd like to close just by acknowledging our collaborators at the New York State Department of Health who have been very involved in our immunogenicity studies, as well as the funding that we've received from the National Institute for Allergies and Infectious Disease, both directly at Sologenics, but also to Dr. Nick Mantis and his colleagues. Thank you very much for your time today.